Firstly, I'd like to acknowledge the traditional custodians on the land we are meeting on today, the Whadjuk people of the Noongar Nation. We pay our respects to elders, past, present, and emerging. So just a few housekeeping. Please feel free to take photos and social media, obviously. The hashtag is ALS MND SIMP, SYMP. So you should all be able to follow what's going on. And please feel free to do that. We will be having some questions at the end, so there'll be plenty of time for a Q&A. We will be live streaming this as well, so if you don't want your photograph taken or you don't want to be part of the live streaming, just move away to the side and that's fine. Hopefully you won't be shot in any of the photographs that people are taking. But first of all, I'd like to ask Drew Bathgate to come up onto the stage. Drew is living with motor neuron disease. He lives here in Western Australia, so we've asked Drew to give a welcome on behalf of Australians, particularly Western Australians, living with motor neuron disease. So we'll let Drew come up the ramp. Thank you. There you go. You all right?
Thank you for your introduction. I was diagnosed with motor neuron disease back in March 2013, so ages ago, and in that time I have been very blessed. What you say, blessed? Well, I know motor neuron disease has a bit of a bad rap, what with all the suffering, and dying, and stuff, but it is through it that I have met some wonderful people. I have 3 lovely and loving ladies, Kylie, Sandy, and Elizabeth, that come into my home and get me going each day. I have my supporters at Motor Neuron Disease Association of Western Australia with Andrew, Leanne, and Sarah, although I am not sure about Sarah, as last I spoke to her, she said she wanted to throw me out of her aeroplane. We love them all, but the one we love to bits is Mary Lee Needham. Mary Lee is my neurologist. I do wonder at times why we love her so much, as she has given me nothing but bad news. You have motor neuron disease, she says. Your symptoms are getting worse, she says. There is no cure, she says. It is not the best way to go, she says, etc., etc. The one I do like is when she tells you to relax, and then comes at your face with a huge metal reflex hammer to the chin. She also does not have a sense of humor, as she now growls at me when the intern goes to take blood, apparently pretending to faint at the sight of blood or sharp needles is not funny. Who knew? At our first meeting, she gave us the bad news and gave us 2 bits of advice. 1. Do not go on the internet to look up what motor neuron disease was, as it was all doom and gloom. The second bit of advice was that we should go and make plans. As it turns out that she was right on both accounts. From her office, my amazing wife Maureen and I walked to the nearest cafe, sat down and made plans for our first journey around Australia. Like most people, we had the idea of retirement and spending our time together doing stuff. The diagnosis had thrown a spanner in the works, and with the prognosis of only 3 to 5 years left to us, we had to do something quickly. The 3 to 5 years came from, you guessed it, the internet. Yet again, Mary Lee was right, and there was nothing but doom and gloom online. I have ignored the internet ever since. We had the safe choice of doing nothing and stay home, but anyone knowing me, I am a safety officer's nightmare. To my surprise and great delight, Maureen said, "Let's not stay home, let's hit the road." At the time I was able to stand and walk with the aid of walking sticks. I was also able to drive. Mind you, I did have hand controls fitted. I think our choice to go touring was just meant to be, when within 2 months, we had found a Pajero and a 20-foot caravan that suited us perfectly, and at bargain prices. We named our van Gert, as in, our home is Gert, by sea, and the vehicle, Trude, so we had Gert and Trude. Gertrude. Had we taken the safe option, we would not have had adventures or experienced things such as watching sunsets on remote beaches without a single person in sight, getting stuck on 80-mile beach at low tide when I shorted out my gopher, and listening to my wife laugh at me until she realized that she would have to push me onto the beach. Didn't laugh too long then. Visit the War Memorial, and be the first person to lay a wreath from a gopher. I also gave the staff there a heart attack when they got me up to the reflection pool level, and I parked right on the edge during the ceremony. And at the end I sat there a while, whilst people around me left, and they were worried I would forget which way to go, and driving to the pool. Little did they know that while I was sitting there, waiting for people to leave, I was having the same thoughts, and was double-checking and triple-checking the drive instructions to make sure I would go backwards, and not forward. I got to drive my gopher around the grand ballroom at Parliament House, until an usher suggested I slow down. I mean, I didn't know that the ballroom had a speed limit, who knew? We ended up doing 2 trips around Australia, and the last one in 2015 was when I stopped driving. From these trips, we had enough adventures to last us. But we did not stop here. With the help of my amazing family and friends, we have done trips to Bali and Singapore, as well as cruises. Can I suggest to those who have disabilities that cruising is great? You can get around with ease, and they get to know you quickly. Mind you, you do stand out a bit, especially in my gopher that seemed to be stuck on a high speed. If you smile nicely at the staff, and play the cripple card, and shrug a lot, they will carry your drinks for you to your seat. With this, it is not long before they go, your usual Mr. Bathgate. That is when you know, it is time to swap bars. But enough about me. I know, carry on I hear you say, and I could, but I must move on. I would like to give a huge thank you to all the researchers, and those who support them. Research is very important, even to those like me who will not benefit from it. I mean, let's face it, if you came up with a miracle cure today, it will be 10 years before all the tests and clinical trials are complete. I appreciate what you have to go through. So I appreciate it takes time, even if I don't have a lot left. I know I am on borrowed time as Mary Lee gave me the bad news back in 2013 and I have progressed in fits and starts. Sometimes rapid progress, and then periods of plateaus. As I see it, I am benefiting from past research. I am on baclofen and Rilutec to reduce symptoms, a big plus as far as I am concerned. As payback to those who went before, I will participate in any research or educational program. I am part of a research project where I answer questions as to how I am progressing, and then a vampire descends and draws blood, usually Mary Lee, another reason I love her. I also volunteer for medical students to poke and prod for their pre-exams review. That has been of great interest and educational to me as anyone else. I knew I was above the average for life expectancy. What I did not know was that I have some classic symptoms and reflexes and some rare symptom combinations, and the attending neurologists would show the students. So I came away with the knowledge that I am an above average, rare, classic subject, a bit like a Rolls-Royce Phantom. Whilst on the subject of being used to teach, or used as a crash test dummy for poking and prodding, do not be backward in coming forward. We are not made of glass, and it takes a lot to offend us. We sufferers are not around forever, and we have to fill our days in somehow. You just have to ask. The worst you can get is a polite no. I think most of us would jump at the chance of helping. I would like to have a diagnosis tool or test developed to speed up the process, as I found those months most stressful of all. It was said that you do not test for motor neuron disease, you test for everything else, and you are left with it. During this process you find out that every organ you have is working fine, there are no abnormal growths. You start to doubt yourself, are you just imagining it? Mind you, if you can identify it by testing something, then you are on a good track to treat it, so I charge you to find that test. Research is not only nice, but it is imperative. Mind you, you do need to be a bit careful when you do have research subjects. I once had a dog who went into a research project, and it killed him. He used to chase cars all day long, and was exhausted at the end of each day, fit as a fiddle. He was selected to take part in a project for smart dogs. Off he went. And 3 weeks later he came home, and we were told he was so smart that they were not only able to teach him how to read, but they were able to teach him to write as well. So from that day on, he would drag a chair out to the front lawn and sit on it, and instead of chasing cars, he would write down license plates. He keeled over dead within 2 months from obesity. On a more serious note, my family and I lost a dear friend recently, Nunzio. He too suffered with motor neuron disease, and rather than sit around waiting to die, he set up fundraising to support research. At his passing, he charged us all to continue the fundraising, and I charge you on his behalf. I am sure he won't mind. For those researching, to continue in your endeavors until you do have a cure. I thank you all for your attention.
Thank you, Drew. That was fabulous, and a fabulous way to start this very important meeting, connecting people with MND, with ALS, to the researchers and the clinicians here today. So first of all, I'd just like to acknowledge the sponsors of today's meeting, the Perrin Institute here in WA, and also the PayPal Give Team grant, which has enabled us to live stream this event. I'd now like to hand over to Merrily Needham, who is going to chair this meeting. Professor Needham is the current Foundation Chair in Neurology, a joint position between Fiona Stanley Hospital, Murdoch University and Notre Dame University, Australia. She was the head of neurology unit at Fiona Stanley Hospital from 2015 to 2019 and is the current head of neuromuscular and myositis research at the Perrin Institute, Western Australia. Thank you very much, Merrilee.
Thank you very much. Now you know a little bit about me, but I actually do have a very small sense of humour, and I'm sure that Drew didn't realise I had the right of reply immediately after him. I think a very important word that he did leave out is when I said there's no cure for motor neuron disease. I meant to say that or I might have said, there's no cure yet for motor neuron disease. And I hope that what we get from the symposium today is hope. And you'll see how hard people are working towards changing the future with motor neuron disease. And it's in fact very fitting that our very first speaker, Professor Matthew Kiernan, is speaking on the topic, Hope is on the Horizon, Advances in Research. For those of you who don't know Professor Matthew Kiernan, he is one of the giants of neurology in Australia and in fact the world. He is the current Bushell Chair of Neurology at the University of Sydney in the Faculty of Medicine. He is also the President of the Australian and New Zealand Association of Neurologists and a Director at the Discovery and Translation Centre at the Brain and Mind Centre in Sydney. Matthew is also the Chair of the Pan-Asia Consortia for the Treatment of ALS, or sometimes known as PACTALS, and it is my great pleasure to introduce him to you all this afternoon.
Well, thanks, Merrilee, and it's a very hard act to follow, Drew Bathgate, but I'm glad that we're in the presence of his classic greatness and his Rolls-Royce approach, and I think that's a good message to bring forward. I'd like to thank Perth for hosting the International Symposium, and it's really great that there's so many patients and families and carers at today's meeting. It's really only through patient advocacy that many of the things that we're going to talk about has really reached fruition, and it's a very strong partnership with patients, their families, clinicians and researchers, and I think that's really been brought together through this international symposium. We have a lot of involvement of patients, plus the basic scientists trying to educate the clinicians and the clinicians trying to at least provide entertainment for the basic scientists. So I think that there's been, massive awareness now in motor neuron disease, and that was really triggered off by the Ice Bucket Challenge. Certainly in Australia, there was $20 million of donation. In the United States, 140 million. In the UK, 40 million. In, Netherlands, Canada. And that really has spurred research into motor neuron disease, and everyone now knows about motor neuron disease, so the community awareness has been great. And it's been taken on by another level through MND Australia and MND Research Institute of Australia, and also through events like the Freeze at the G with Neil Danaher. And basically, one day at the MCG, seeing people walking around with MND hats, generating 1.2 million just from selling beanies, generating $6 million of support for MND research in Australia is really powerful. And it brings a lot of people into the discussions, including Greg Hunt and politicians in general, state ministers. And that's really where we can try and push things further. And this is walking from Federation Square to the MCG, everyone wearing MND beanies, and you wouldn't have imagined this, 5 or 10 years ago. So the whole landscape has transformed. So Australia actually has a long tradition in MND research, and it goes back to A.W. Campbell, and he was an Australian working with Sherrington in the late 1800s, early 1900s. And his critical part was to actually suggest that MND was a disease of the brain. And that's been a very controversial area. Most people have got into motor neuron disease through neuromuscular medicine, but more and more, particularly through some of the more recent discoveries, we've realized that MND is a primary neurodegenerative condition, and it very much has its onset and origins in the brain. The other thing is, so more than 100 years ago in the Medical Journal of Australia, here is a case report detailing motor neuron disease, and I think it gives a very classic history. So typically it never experienced a day's illness. It's a bit like the dog we heard who's chasing the cars. He's very fitness orientated, then developed right hand weakness in the muscles between the thumb and the index finger. And again, that's a very typical beginning for motor neuron disease. And why is that? Well, I think this is really reflecting the evolution of humans. We're the only mammals who have a pincer grip. So this has been very important in terms of like moving slides along, holding credit cards, using knives and forks, etc. But that is a massive design flaw in the human brain. It's got a huge amount of energy needed, and that's often the other area that we've evolved, obviously, is communication. Despite Drew's dog, the fact that we can speak and interact with each other, again, is an evolutionary area, and I don't think this is pure chance that these are the areas that are usually selectively affected in motor neurone disease. And this presentation we can work out now from 100 years ago is what's called the split hand of ALS. It's like there's been a split down here, and the reason for that is these areas here have got massive representation in the parietal cortex. So this is an upper motor neurone presentation. So that's a brain presentation of motor neurone disease. Then they describe very similar changes on the left-hand side. So typically, if you have involvement of your dominant right-hand side, it'll move very quickly to the same muscle as the left-hand side. And that's presumably through transsynaptic movement across the corpus callosum, which is the part that joins the two hemispheres together. So we have a part of the brain that stops us, simpler mammals, when they do one task with an arm or a leg, the other side does exactly the same because there's free communication from both sides, but human evolution has triggered off this process, and ultimately the diagnosis is ALS. So I think similar to the past sort of giants of neuroscience, we've been very keen in Australia to have involvement with the global approaches, and we'll hear of many of those approaches during the week, but the two speakers here this afternoon have driven many of those, and including Project Mind, trying to get all of the genetics for sporadic ALS patients, 12 or 13,000 individuals. And it's only through these powers of large international consortia that we can have any chance of trying to work out this condition. If it was a simple condition, one gene leading to one disorder, it would have been solved a long time ago. But it's a very complicated multi-hit process. And we've been very grateful for the support from MND Australia and Carol Burks with Project Mind. And already, by being part of these international consortia, we're seeing that new understandings of the genetic architecture are coming out, and also the links across brain and mind diseases. So patients who have motor neurone disease have higher incidence in the extended family of people with autism, bipolar disorder, schizophrenia, Parkinson's disease. So this is really part of a sort of a global brain and mind series of conditions. And what we've tried to do in Australia, as well as contributing to the international consortia, is develop our own national pathways, and very much with Merrily here in Perth and our colleagues in South Australia, Queensland. So we're trying to now generate, it's called a partnership. So we have an app that is patient-driven, so the patients have all of their information, seeing the various clinicians that are involved and allied healthcare, etc., but at the same time driving the research and availability into clinical trials, providing samples to our Australian consortia, which is driven by Naomi Ray in Queensland. And we're starting to see now very much a more sort of general involvement and a more focused approach that is already leading to better results. But in terms of treatment, I suppose the biggest sort of step forward from when Charcot had described the disease occurred in sort of the late 80s and 90s, understanding the genetics of motor neurone disease. And the first mutation that was identified was superoxide dismutase. And in fact, a number of Australian families contributed to those findings, particularly through Garth Nicholson and Nigel working here in Perth. But really, I suppose it was a bit of a surprise because no one actually knew what SOD1 did. And they went back to basic biochemistry and tried to understand how mutation in that gene would lead to the process of motor neurone disease. And that led to people looking at it's part of a redox reaction, excitotoxicity. Glutamate is involved, and in fact, colleagues in the north of the UK, so Pam Shaw, who'll be at this meeting, and Jeff Rothstein in America started measuring cerebrospinal fluid in patients and found that glutamate was upregulated, and that led to this idea of excitotoxicity, that something triggers off the motor neuron and it starts firing, releasing glutamate, and glutamate is toxic, and that leads to the cascade of cell death. And I suppose very soon after that, riluzole was identified and went through clinical trials, driven particularly through Vincent Meninger from Salpêtrière. And that was a way of trying to reduce glutamate, or so it was thought. And these studies were published in The Lancet and New England Journal. Australia was involved in those clinical trials, and we were a clinical site for the introduction subsequently of riluzole. But it was a simple idea that maybe riluzole reduced glutamate. But really, I suppose what we're starting to see is maybe riluzole is slightly more complicated. And rather than getting into too much sort of research, but if you stimulate the motor neuron with a magnetic stimulator, patients with motor neuron disease have a hyperexcitable cortex. And that probably leads to symptoms like cramp, fasciculation, and so on. And when you start patients on riluzole, you start to see that the hyperexcitability is reduced, so inhibition is increased. It doesn't actually get back to normal, but it starts to approach normal and then gradually winds down. It's also become clear that riluzole works through persistent sodium conductances at a neuromuscular junction. And that led to a whole restaging of what riluzole may do, and that was undertaken by Amar, who's the next speaker, working through his King's database, showing that riluzole has positive effects in motor neuron disease patients at all stages of the disease, including the last stage, the end stage of the disease. And probably that suggests that it may be working through different pathways to maintain function. So that's something that we need to consider further. In terms of therapies, really it's directed by being part of a centre that has an interest in motor neuron disease, and ideally multidisciplinary care. So this is our team in Sydney, which has a nurse coordinator, representatives of the association, occupational therapist, speech pathologist, physiotherapist, social worker, and it's through a sort of a combined team that outcomes are better for patients with motor neuron disease. And this was a study published by Orla Hardiman, who will also be attending the meeting, looking at her clinic in Dublin, and showing just by attending a multidisciplinary clinic, your survival was increased by 300 to 400 days relative to attending a single neurologist. So that's an important message. And then if you add riluzole to multidisciplinary care, the outcomes are much better. Separately, we're understanding now the role of non-invasive ventilation, so putting patients on ventilatory support, particularly overnight. And this was a study, a trial undertaken by Pam Shaw and her colleagues in the north of England, and they showed that non-invasive ventilation improves survival, also improves quality of life. So patients feel better, they have more energy, less headaches, and more active. I suppose what we need to do now is work out when do we institute non-invasive ventilation. As it stands, we start the therapies usually when patients are symptomatic or when their respiratory function is deteriorating. The other thing to say is we've actually got a lot of therapies for motor neuron disease to cover the symptoms, so muscle cramps, spasticity, overproduction of saliva, breathlessness we've covered. We're understanding now the critical role of weight loss and nutrition, and also emotional ability. And what we've tried to do on a national platform, and this was published in the Medical Journal of Australia, is get all of the clinics together to try and have a national approach to therapies. Merrily mentioned Pactals, and that's been an exciting development. So although we appreciate our colleagues in Europe, they are really probably the dying force. And the strength of the region is going to be in China, India, with $1 billion each. So Leonard has come down to a mission and he wants to take over. But we've always said, look, we're going to stand it alone and see what we can do. But actually, it turns out there's great strength in Korea, Japan, and we're trying to set up now a trials consortia to drive these approaches together and also to better understand the genetic differences. And we've had our first standalone meeting in Korea, and our next meeting next year will be in Japan. We've also taken on some of the concepts that have been developed by Amar and Leonard, looking at motor neuron disease as a multi-hit hypothesis. So we've now been able to look at it in terms of Australia, but also now with 5,000 Japanese patients, 3,000 Korean patients, and shown that motor neuron disease is a linear relationship, suggesting that there are 6 hits that have to occur for motor neuron disease to develop. 3 of those hits, if you come with a family history of motor neuron disease, that would account for 3 of them. And then what are the other 3? Well, we're not really sure, and they could be random. But this is where we really need to get together in larger consortia. There are some interesting studies coming out now as well for new treatments, and one of the areas that is taking off is the whole role of inflammation in neurodegeneration. And this was a release from AB Science in France for their medication tyrosine kinase inhibitor masitinib. And this works through trying to reduce inflammation and microglial activation. And there's going to be a larger scale trial undertaken in which Australia will be involved hopefully next year. Now, while riluzole was approved in Australia after the trials in the mid to late 90s, the second medication to get FDA approval has been edaravone, which is owned by Mitsubishi Pharma. Again, works through free radical scavenging. And at the moment, patients who want to access that have to go either to Japan or work out some import license to get it back here to Australia. Although I think that there's going to be some announcements shortly that there will be a company in Australia that will be releasing it, so edaravone should be made available to patients around Australia. Courtesy of our National Clinical Trials platform, we've been able to now take on investigator-driven studies as well. And this was a study that was supported through the Medical Research Futures Fund. It was a $4.5 million donation so that we could look at Tecfidera, which is a therapy registered for multiple sclerosis, trying to alter the populations of neuroinflammatory regulatory T cells. They weren't attacking the motor neuron, so they may slow down the process. And we recruited 120 patients around Australia with sites here in Perth, Brisbane, Adelaide, Melbourne, and we're just about to lock the data down and see what those results show. At this meeting later this week, we'll hear from Cytokinetics, who are going to explain the results of their Fortitude study. There's a lot of interest, obviously, in genetic therapies, cell therapies. So the SOD1 ASO, anti-nucleotide sensing oligonucleotide study, has commenced in Australia with Westmead Hospital and Steve Vucic as the national lead. We'll be doing C9-ORF studies next year. There's also a press release today for the results of Neurone, so a stem cell approach that was driven particularly through Massachusetts General Hospital, showing that there may be some benefit in a phase 2 study for those who have very fast progressing disease. Ryuji Kaji from Japan will be here at the meeting. He'll be presenting his findings on methylcobalamin. He's now started a phase 3 study to get FDA approval and Japanese approval for high-dose methylcobalamin. So this is a process that is moving forward. Another inflammatory approach has been driven through Australia. So Julian Gold did the Lighthouse study. So that was a sort of a proof of concept and a safety study looking at Trimec, which is a medication for HIV therapy, and showed that firstly it was safe, it didn't affect riluzole, and had some suggestive movements in the right direction in terms of biomarkers. And now we need to do a larger phase 3 study, which will hopefully be a collaborative approach through Europe and Australia. I suppose in terms of not trying to have excuses, but it's taken us a while to actually understand, and we haven't fully understood the biology and the pathophysiology of the disease. And without understanding the disease, we still inevitably have to have sort of shots in the dark and try and do approaches without necessarily having the full biology. The other thing that we're working out is that not all ALS or MND is the same. So there's a lot of heterogeneity in patients. And there's also a lot of differences in how people respond to the disease. Some people may live for 10 to 15 years. Some people are dead within a few weeks. So there's a massive range, and we don't have good ways to try and identify that. Although by trying to get to the genotype and then the resultant phenotype, we may get some better understanding. We've also realised that all clinical trials from now on should have a biomarker component, so that if the study is negative, at least we're going to learn more about the biology of the disease itself. And I suppose, we've also been involved in tricky areas as well. So dexpramipexole was a study that showed benefit at a phase 2 study. So Biogen invested $40 million to do a large-scale international trial with Australia as one of the sites, 990 patients, and then we got to the end of the trial to find out that it was negative, and that's sort of a really flattening experience. And part of that is, again, not knowing the biology, but also trying to interpret phase 2 early phase data. So the other thing that we're getting a better understanding of is that there are, pre-morbid personality types and physiques, and there does seem to be a higher incidence in people who are very physically active. And this was really brought out firstly by Adriano Chio, looking at the football players in Italy. We can see and Lou Gehrig himself, but on the right-hand corner up there, you can see the Australian Davis Cup squad, so Brad Druitt, who looked after had bulbar onset motor neurone disease. The person beside him, Peter Doohan, who recently died of motor neurone disease. So 2 players in the one team getting motor neurone disease. We've got Just van der Westhuizen, the captain of the South African football team, Neil Danaher. So there seems to be a suggestion that there are different types of protoplasms linked to motor neurone disease, and this is a very typical history. You know, this is a patient from Northern Territory, and the background is there's no medical background. So these patients don't present with any past medical history. They've got no medical notes. Whereas when you see someone, for instance, who has a stroke, they might have multiple admissions with diabetes, poorly controlled blood pressure, heart disease, etc. And going back through this, generally motor neurone disease patients, they have a low or normal body mass index. They're rarely obese. They're usually fitness-orientated, athletic, and studies from both the UK and also Leonard's group have shown reduced coronary artery disease in patients and their relatives. And some people think that there is also a personality type with motor neurone disease. So certainly we find our patients are very, they say hello to us, which is probably more than our own colleagues do in the department. So there's something different. So we've started looking at our patients and going back with their carers and asking them, you know, did they notice any changes? And in fact, 3 or 4 years before they developed motor presentations, their carers had noticed that there'd been, you know, changes in their personality, sometimes more impulsive, more sort of prone to temper outbursts, some people more sort of apathy. So there'd been some cognitive changes that had presented well before the motor presentations. And similarly, there seems to be metabolic changes. So oftentimes patients will lose dramatic weight loss, 20 to 30 kilos, before they've had any bulbar weakness. So there's some sort of metabolic process linked to the degeneration. And these are studies that we've been taking forward in Sydney, but also colleagues in Brisbane, and looking at insulin resistance, lipid profiles, and eating behaviour. And it's very interesting, when patients who have a normal or low BMI start to increase their BMI, that's often the indication that they are developing cognitive problems and potentially frontotemporal dysfunction. So I think the other area that we're very interested in is we see patients typically at this region here, and it's very hard from once you diagnose them over, say, a 12 to 18 month period to really find major changes. But we don't really know what has gone on during the course of their adult life, going all the way back actually to conception, in utero, delivery, the maternal environment. And this is, I think, where there's going to be a lot of important discoveries going into the next few years. We also don't know when the process of neurodegeneration is triggered off and how that is progressing, because we don't really have a marker of these changes in the brain. And so the other thing to say is that we do know that there seems to be a focal onset, and should we be trying to direct our therapies very early on at that center in the brain? And I think with the advent of nanotechnology, drug delivery systems, I think we're going to start seeing some better outcomes for our patients. And so on that note, I would say that I think there is hope on the horizon. I think the whole landscape of motor neurone disease is changing. We've been very fortunate to have great community involvement, patient advocacy, great generation, basically great donations and philanthropy, and that is really driving now. We've got industry, philanthropy, government ears, and I think that we're getting a much better understanding of the biology of motor neurone disease, and I think the outcomes are already better, and that was established through Stan Appel, who's been running a clinic since the 1960s in America, and he's shown that the outcomes now are much better than the patients for 1960. So of course we want an even greater acceleration and improvement, but I would say that a lot of these things are well underway, and I'll leave it at that. Thank you.
Well, thank you, Matthew. I hope you all feel really uplifted by that. Our second speaker is Professor Amar Al Chalabi. He is the Professor of Neurology and Complex Diseases and the Genetics Head at the Department of Basic and Clinical Neuroscience and the Director at King's Motor Neuron Disease Care and Research Centre in London. Professor Al Chalabi will discuss causes of motor neuron disease, which I'm really looking forward to hearing how to answer that. Always the question, why me? Why has this happened? So welcome.
Thank you, Marilee. So I will be talking about what causes motor neuron disease. And one of the things I'd like you to remember while I'm giving this talk is that a lot of things are associated with motor neuron disease, but association doesn't mean causation. So there are two very obvious causes of motor neuron disease: family history and age. We know that because about 5 to 10% of people, when they come to clinic, have a family history in a first or second degree relative of motor neuron disease. And that tells us that because there's a familial component in at least some people, there must be a genetic component in at least some people. The other aspect is age. We know that the older you get, the more likely you are to develop motor neuron disease. Typically, the age of onset is in the 50s or 60s, but actually the age of onset is about 70 in population registers. And that's because the people who come to clinic are generally younger and more motivated. So the actual age, when you do the proper studies, shows that the risk just keeps increasing as people get older. And that tells us that the mechanism is something to do with aging. So I'm just going to deal with the first of those aspects. So how much do genes contribute? We can try and answer that by looking at twins. Identical twins share all of their genetic information, whereas non-identical twins only share half of their genetic information. And both types of twins share a lot of their environmental data, environmental components. So we can look at identical twins and ask, are the same things happening to both twins? If they do, if that happens more frequently than in non-identical twins, that tells us there's more likely to be a genetic component because they share more of their genes. So we've looked at about 170 twin pairs. And in those twin pairs, if you look at the identical twin pairs, in about 5 of those twin pairs, both of the twins develop motor neuron disease. Whereas in 44 of those twin pairs, only one of the twins develop motor neuron disease. And if we look at non-identical twins, then never did both non-identical twins develop motor neuron disease in our group, whereas in all the 122, one of them was affected, because obviously we selected them for that reason. So if you do the maths, that tells you that about 60% of the risk in developing motor neuron disease comes from genetic factors. Now, these weren't twins with a family history otherwise of motor neuron disease. So that's telling you that there's a genetic component for motor neuron disease in general. But why don't we then see a family history in everybody? Why is there a genetic risk without it being inherited? That's because most genes only have a small effect. So this is what's called a Genome-Wide Association Study, or a GWAS, where you look at about, in this case we looked at 40,000 people, 13,000 people roughly with motor neuron disease, and about 25,000 people without. And then we looked at about 17 million different genetic variations in the genome. And we tested each one to see, is a particular genetic variation more common in the people with motor neuron disease or more common in the people without, or is there no difference? And then we plotted that on this graph, and every single dot represents one of those tests. And the higher up on the y-axis that the test appears, the more likely there is that there's a difference between the two groups. And what you can see, all the red ones are where we've seen a genetic signal. There's one very big one, which is the C9orf72 gene, which I'll talk about a little bit in a moment. But you can see there are lots of hits. And that tells you that there are lots of genetic factors that are all contributing. Now, each of these hits will only contribute perhaps 2% or maybe up to 20%, but usually about 2 or 3% to your risk. So it's not a very, very big increase. And that's why we can have a genetic component without it being inherited. It's just telling you those genes are slightly increasing your risk, and they all add up. If you happen to have many of these gene variants, then your risk might have gone up by 25, 30, 40, or 50% compared to the general population. But you're not going to transmit all of those genes to the next generation, because we only transmit half of our genes on average. Now, some people do have a family history, as I pointed out, and that's because some genes have a very big effect They really increase your risk greatly. These are some of the genes that have been found over the last 25 years. You can see that the rate of gene discovery is accelerating. Actually, this slide is about 2 years old, and I haven't updated it because I'd have to add about another 15, 20 genes. The rate of gene discovery is doubling every 4 years. These are for the big effect genes. What I've done here is the graph shows the size of each gene in terms of its importance. So you can see there are 4 particularly important ones on this graph, and they're important because people who study motor neuron disease think they're important, not for any other reason. So SOD1 is the first gene discovered. That's the one at the very left at the bottom. That's important because it's the one we've studied the most. For many years, it was the only gene we really knew had anything to do with motor neuron disease. Then 2 others, TARD-BP, which codes for a protein called TDP43. That protein is found in nearly everybody who has motor neuron disease in their nerve cells, regardless of whether they have a mutation or not. Only 1% of people have a mutation in the gene for that protein, and yet nearly everybody with motor neuron disease has it accumulated in their cells. We don't understand why that is, but it tells us that it's something to do with the disease process. Then there's another related gene, FUS, which tends to cause a very young onset, aggressive type of motor neuron disease. And then the big circle you can see in the middle is for C9orf72. That's a disease gene that, even in people without a family history, can account for up to 8% of the cases we see. Actually, I'm just going to tell you one other thing about those genes. So they're color-coded. You can see that there are blue, red, or green labels. The blue labels are for genes that are to do with the cytoskeleton. That's the skeleton for the nerve cells, because nerve cells are a very strange shape. They're basically a circle with a very, very long tube, and that tube is microscopic, but can be up to 1 meter long. So it's an extremely strange shape, and it needs a strong skeleton to allow that to happen. And several of the genes are involved in the cytoskeleton. The other group you can see is RNA processing genes. RNA processing is the step that your body takes to take the genetic code and make it into you. It's the translation step to take your genes and make them into proteins. So that seems to be important for nerve cells as well. And then the final group is the proteosome genes. The proteosome is the part of the cell that gets rid of damaged proteins. It's like a dustbin for the proteins. So those 3 processes seem to be important for motor neurons. The cytoskeleton, translating the genetic information into proteins, and getting rid of damaged proteins. So the genes have told us that those 3 things are important. Now, some of those genes, as you've heard already from Matthew, are involved in other conditions than motor neuron disease. So, for example, the C9orf72 gene that I told you about, in some people it causes frontotemporal dementia. In other people it causes motor neuron disease and frontotemporal dementia, and in many people it causes nothing. In a few people, it increases their risk of Parkinson's disease or schizophrenia. Other genes also can increase your risk of frontotemporal dementia, such as TARD-BP and FUS. Some are involved in another brain disease called ataxia, and some are involved in Parkinson's. And there's also a 14% overlap between the genetic risk factors for motor neuron disease and the genetic risk factors for schizophrenia. So that's an unusual thing to know. Why should motor neuron disease genes also increase your risk for these other conditions? So we can conclude at the moment that one cause of motor neuron disease is your genetic makeup. But it's not clear why there's so many subtypes. Why isn't it just one gene that causes motor neuron disease? And why is there an overlap with these other conditions? So the second obvious cause I told you about was age. So this is a graph of the likelihood that you'll develop motor neuron disease by your age. You can see that your risk just keeps increasing as you get older. You can also see that men always have a greater risk than women. So the ratio is about 3:2 for whether men develop motor neuron disease or women. And that's true even if you've got a disease in your family, a disease gene in your family, which is strange because you have a 50-50 chance of inheriting that disease gene. So for some reason, women are protected in some way from developing motor neuron disease. The other striking thing on this graph that you can see is that by about the age of 85, you've got a 1 in 300 chance of having developed the condition. That's much higher than most people realize. That's your lifetime risk. So most people talk about the incidence of motor neuron disease, which is an epidemiological thing. It's a statistical, technical number. It's the number if you observed a standard population for 100,000 years of one person, or you observed 100,000 people for 1 year, What's the probability that someone will develop motor neurone disease? It's a very difficult number to understand. Whereas your lifetime risk is a very easy number to understand. It's just a chance when you're born that you will die with motor neurone disease. What's that chance? It's 1 in 300. So that means that at a symposium like this, about 3 of the delegates are likely to develop motor neurone disease. That's about the same as for multiple sclerosis. But the reason why multiple sclerosis, actually in the UK, because I don't know what the incidence is in Australia, but I'm assuming it's something similar. The reason multiple sclerosis is more frequent is because it doesn't usually affect your life expectancy, whereas motor neurone disease does. So why is age important? I'll leave you to think about that for a bit. Environmental risk factors are the other big search area in motor neurone disease. Now, whereas genetics might seem a very difficult problem, because we've got 24,000 genes and it's very, very difficult to spot one genetic change in a person, environmental risk factors might seem easier, but actually it's much more difficult because the search space is infinite. And it's not just infinite in terms of the number of environmental things you'd need to ask about, it's also extending over somebody's lifetime. And nobody can remember things from when they were a child, or a potentially simple thing like maybe you knocked your knee when you were a child and it happened to get a particular type of dirt in it or something like that. It could be something really trivial that you'd never know. So the search space for environmental factors is very difficult. And as a result, despite a lot of searching, we don't have any reproducible associations. We've got lots of strong likely associations, and I've listed some of them here, but nothing has been convincingly, definitely shown repeatedly time after time in every single study. So the ones that have the most evidence are military service, particularly if you're deployed in the Second World War. That seems to have increased risk by up to 7 times in some studies. Any place, I'll show you in a little bit. Any lifestyle, smoking is in some studies a risk factor, in other studies it isn't a risk factor. Probably the balance of probability is that smoking is a risk factor, but it's not definite. Exercise or sport is a very difficult one. You heard from Matthew that there is a lot of association with sport. That doesn't mean it's causative. In the same way as drowning and ice cream sales are associated, it's not because ice creams cause people to drown. It's because hot weather makes you makes you eat ice cream and also makes you go swimming. And so the two are associated. So exercise might be associated with motor neurone disease through a third thing in that way. It might also be that, for example, exercise might make the disease slower. It might be protective once you've got it, but have no effect on risk. Now, if that happens, then more of the people we see in our clinic will have been people who are sporty and exercise. It won't be because it's caused it, it's just because they're around longer because it's protected them. So it's a very difficult thing to understand whether sport really does increase your risk. And it may be that people who have a sporty motor system are more likely to get motor neurone disease just because having a sporty motor system is more difficult to maintain. A bit like a Ferrari is a nice sporty car, but it's more expensive and difficult to maintain. In terms of football, which you heard about from Matthew, there was a study in Italy that showed that if you're a professional footballer, you've got about 6 times the risk of motor neurone disease as the general population. So that sounds quite a big effect. In fact, if you do the same study and analyze the statistics a different way, it shows there's no increase in risk. So again, it's a bit controversial, and we're just in the process of trying to repeat that study more globally. We reported 3 footballers who were all amateur footballers, grew up in the same village in Kent in the southeast of England, and all developed motor neurone disease at the same time. 2 of them died on the same weekend. So we reported that because it happened at the same time as this paper on football and motor neurone disease came out. In fact, a 4th person who was in that group of amateur footballers who were all friends playing in the same team at the same time, a 4th person has now developed motor neurone disease. So you might think, well, that's really very strong evidence that football is important. But actually, they all grew up in the same small village in Kent. So maybe it's something in that village that's the risk factor. They're all exposed to the same chalk dust at school and things like that. They all played on the same pitches. They happened to all be electricians, and they'd all had electrical shocks. They all had diabetes. They were all smokers. So we're focusing on football because that's the thing we're interested in, but it doesn't necessarily mean it's football. So that's just an example of how difficult it is to really know how to find an environmental risk factor. Life events such as infection might be important. Electric shock, some studies show it's important, others show it isn't. And physical trauma such as broken bones seem to be at a slightly higher level in people who've then got motor neuron disease, but it's again not clear if that's a definite link. Here's a map of every single person with motor neuron disease over a 10-year period in the southeast of England. So the black dotted line is the border of the study. At the top, it's bounded by the River Thames and by the sea, and along the south and east, it's bounded by the sea. So I don't know if you can see any clusters yourselves. Can anyone see any? Yeah? So there's some around London. There's some around the east tip of Kent. So I've put circles around the ones I see. Now, the problem is, those are just circles that I can see. Now, I told you motor neuron disease is a disease of increasing age. More people in the UK retire by the coast. And so you're going to get clusters by the coast because it's just an older population. Why is there a big cluster in London? Just because more people live in London. Okay? So seeing clustering like this doesn't necessarily tell you anything. You have to take into account all of the other factors and account for them statistically. If you do that, most studies do show a small increase in risk based on where you live or a decrease in some areas. But it usually only increases your risk by maybe 20 or 30% at the most. So there might be something in the soil, in the air, electrical pylons, we don't know, that increases risk for some people. What about viruses? Well, poliovirus causes a motor neuron disease. It causes weakness. Do other viruses? So we and others have looked for that. In fact, other enteroviruses like poliovirus don't seem to cause motor neuron disease of the classic type that we're talking about. But if you look at retroviruses, about 59%, 60% of people with motor neuron disease have a retrovirus signature in their blood, whereas only 5% of the general population does. If you look at relatives, then they also, about 60% of relatives of people with motor neuron disease also have this signature of a retrovirus in their blood, whereas the spouses match the general population. Only about 5% of spouses do. So that tells you it's an inherited pattern of disease, not an infective pattern of disease, which is a bit strange. How can you have an inherited virus? Well, as it happens, you can. There are things called human endogenous retroviruses, which are ancient viruses embedded in human DNA through evolution. They're in the same group as HIV, and in fact, HIV tries to embed itself in your genome if you're infected. So these aren't infective. Everybody's got them. Everyone in this room has many, many HERVs in their body. In fact, 8% of your genome is comprised of these human endogenous retroviruses. You can contrast that with just 1% of your genome that codes for you. So 8 times more of your DNA codes for viruses than does for you. Actually, they've now become so much part of us that they're essential for some things. We use HERV-K, which is one of these retroviruses, to actually form our placenta. So human placentas are formed with this virus. They can't form without it. HERV-K in one study has been found to be a possible culprit for motor neuron disease. It's toxic to motor neurons. If you have mice that express HERV-K, they develop motor neuron disease, and if you put HERV-K into human cells in a dish, then they don't like it. They die. But that would make it a genetic cause, not an environmental cause. There's also the internal environment, the gut bacteria, which seem to influence the immune system. And in people with motor neuron disease, there is a reduction in a particular group of bacteria that produce a chemical called butyrate that your gut cells like. There's a reduced level of that. And the immune system also seems to be important. There's a type of police immune cell called the T regulatory lymphocyte, and that controls how aggressive your immune system is. And in people with motor neuron disease, the levels seem to be lower than they should be. So it may be that controlling your immune system is an important treatment avenue. So it's difficult to find environmental causes as well. Why is that? So I've given you a few mysteries there. So the mysteries are, there's an increasing risk with age. There's no single cause for motor neuron disease. Environmental causes are difficult to find. One gene variant can cause different diseases. Not all gene carriers develop motor neuron disease. I told you about people with C9orf72 who never develop motor neuron disease. It seems to start in one region, as you heard from Matthew, and then spread, and it's specific to motor neurons, but it can include others. So why is all this the case? We can try and learn from cancer. In cancer, we know it's a multi-step process. So you have something like this. You inherit some cancer genes, which are mutated in you. Maybe you smoke, which mutates a few other genes. You might then have a viral infection that activates your immune system a particular way, and then a bacterial infection that activates it again, and maybe you drink a lot of coffee, which activates your cells another way, and then you develop cancer. Okay, these aren't real examples, but they are typical kind of things that you might hear about as increasing your risk. So we can ask, is motor neuron disease a multi-step process like cancer is? So the answer is it's quite easy to tell, because you can use a simple mathematical relationship, which I can explain to somebody afterwards if they want. But basically, if you do a graph of the log of the incidence of motor neuron disease against the log of the age at which you're measuring the incidence, then if it's a straight line, that means it's a multi-step process. And the slope of the line happens to tell you how many steps there are. So we did this in our Southeast ALS register in the UK, and it looks fairly like a straight line. The R² number there, you can see, 0.95. The closer that is to 1, the more like a straight line it is mathematically. And 0.95 is a very, very high number to get. So we then went to our colleagues in the Netherlands, actually to Leonard, and this was the result from his register with an R² of 0.99. In Italy, it was 0.95. In Ireland, it was 0.99, and in Scotland, it was 0.97. And when we put them all together, it was still 0.99, which tells you they all had the same slope. And actually, as you heard already from Matthew, it's been confirmed in an Australian register, and that too has the same slope. So it looks like motor neuron disease is a multi-step process, and if you look at the number of the slope estimate, it's actually always around 5, and that's always one less than the number of steps you need. So that tells you that motor neuron disease is a 6-step process on average. We don't know what those steps are, but it tells you there are 6 molecular steps required to develop the disease. We know that genes can account for some steps. So if you happen to carry the C9orf72 gene mutation, then that'll account for 3 of your steps. If you carry the TDP43, that'll account for 2. And if you carry SOD1 mutation, that accounts for 4 of your steps. So you only need 2 more to develop the disease. So that's why those genes have a big effect on your genome. That's why they greatly increase your risk, and that's why in those people, there's often a family history of the disease. They don't have many more steps to get to develop the disease. And this multi-step model explains lots of these mysteries. The increasing risk with age is because you need time to develop all these hits. The fact there's no single cause is because lots of different things might cause the hit. In the same way as smoking can increase your mutation rate, so can having an X-ray or traveling in an airplane or living in a radiation area. Environmental causes are difficult to find because lots of different things can cause the same outcome. One gene variant can cause different diseases, because if you go through steps 1, 2, 3, 4, 5, 6, you get motor neuron disease, but if you go through 1, 2, 3, 4, 5, 7, you get frontotemporal dementia, for example. So if you go down different pathways, you might get different diseases. And the reason not everyone carrying a disease gene will develop motor neuron disease is because if they don't have all 6 steps, if they don't have the last ones, they won't develop the disease. It starts in one region because only motor neurons are, because only one group of cells will have been affected with all 6 steps to start with, and it's specific to motor neurons because motor neurons must have something that's vulnerable for one of those steps, at least. So the multi-step model explains quite a lot of the reason why people get motor neuron disease. How can we put all this together? Well, we can think of disease like this. So you've got your burden of disease-causing factors and your health, and you're born healthy. You're born with a particular amount of genetic load, and then with time, you get cell damage and you have environmental exposures. But unless you reach a particular threshold of burden of disease, you're not going to get it. So, GTE, genes times environment. If you do have enough of an exposure, then you'll have motor neuron disease, and then you have a self-perpetuating process. And you can imagine the 6 steps along the bottom. So now you can see if you've got lots of the small genetic risk factors, they might account for 1, 2, 3 of the steps. Or if you've got one of the big genetic factors, that might account for a lot of the steps. And it also tells you that if you want to find environmental risk factors, actually what we should probably do is look at people with, for example, SOD1 mutation, because they've only got 1 or 2 steps more to find. Whereas if you're searching in everybody with motor neuron disease, it's going to be more difficult. It's a much larger search space. So just to conclude, motor neuron disease seems to be a 6-step process. Some of the steps can be genetic. It's not easy to find definite environmental risk factors. It looks like military deployment might be important. Sportiness might be important, doesn't mean it's causative. And smoking might be important. And it's likely to be a combination of genes and environment. And by environment, I mean both the internal and the external environment. So these are some of the people who've sponsored our research. And I'd like to thank all the patients who gave their time to our research, without whom it's impossible. And this is my research team that did a lot of the work you've seen. Thank you.
Well, thank you very much. I think I have a much better way to answer that question next time I'm asked it in the clinic. We're going to move to our final speaker, and then we're going to open the floor for questions. So please start thinking about what questions you'd like to ask these world experts in motor neuron disease, because we're going to get cracking with question time straight after Professor Leonard Vanderburgh. But please allow me to introduce Professor Leonard Vanderburgh. He's the Professor of Neurology and the Director at the Netherlands ALS Centre University Medical Centre in Utrecht. He's the Chairman of the Neuromuscular Centre in the Netherlands, and the chairman of the European Network to Cure ALS, NCALS, which hopefully will partner with our Pan-Australian one to really get a global effort towards eliminating ALS. Leonard will discuss clinical trials, accelerating ALS or MND therapy and development through new innovative clinical trial design. Thank you very much.
Thank you very much for the invitation to be here. Speak here. I thought after a 24-hour flight, I come over one day early to see Perth. But then Carol asked me to speak here, and saying no to Carol is suicide. So I'm happy to be here, and it's good reason to come back to Perth another time. Treatment. Why does it take so long to find a treatment? And I understand that you're all very impatient and you're short of time. And we are also, as researchers and clinicians, impatient because we see lots of patients and we do want to find a treatment for our patients. Why does it take so long? It's because the process is quite long. The first part, you may have seen this before, is the preclinical part, where all the research is done in animal models or cellular models. And then if there's an interesting candidate treatment, it needs to be tested in healthy individuals, whether it's safe. That's called the phase one study. It also may involve some patients to get some feeling about safety mostly, but also about dosing of the treatment. The next phase is phase two. That's a larger study. It involves ALS patients, and it's designed to investigate whether the drug is safe and maybe some clue whether there's some effect. If that is true, then a next phase will start, a phase 3 study, and that's a larger study, and that's all aimed to investigate the efficacy of the drug. So that whole process may take years, up to 10 years. So what our goal is, and what we should do, is try to accelerate that process. And I'm happy to say that it's actually time for some optimism. Many discoveries in the preclinical phase have been made, and there's now also a success in gene therapy showing that ALS is a treatable disease. So that's very important. It's a first step, but hopefully many steps will follow. So we have to concentrate on the phase 2 and the phase 3 studies to accelerate those, to accelerate the discovery of a treatment. This may be a little bit busy slide. It's just an overview of the phase 2 and 3 studies that have been done over the last decades, and it shows that if you, the one on the left is the time of the trial, so the duration of the trial. And as you can see, there's quite some variation. I think that's the message of this whole slide, that between all these trials, there's just lots of variation. So one trial lasts only several months, while another trial lasts 18 months. And that's also true for the primary outcome. Now, primary outcome means the primary function or the primary measurement that will determine whether a study is positive or negative. So that can be a functional score or survival, but also muscle strength measurements. So there's quite some variation also in the primary outcome measures and also in the sample size. So as you can see, the phase 2 studies are smaller and the phase 3 studies are larger. But even between the phase 3 studies, there's lots of variation. So if we could find what we call a master protocol, one protocol that fits all drugs, that will help enormously in accelerating the process. So we have to work on that and to find consensus among researchers, but also companies, to have a more harmonized drug discovery design. So what is the aim of a clinical trial? Well, one aim, of course, is very important. It needs to be a safe drug. So that's the goal of the aim of all phases in the process. But what the second aim, and that's even more important, is that it should slow down the disease. And how do we measure that? That's very important. So how do we determine whether a therapy is effective? So if there's a large effect, so this is an example of antibiotics, you don't need a very long trial to investigate the effect of antibiotics. There's a good biomarker. As you can see here, you measure the growth rate of the bacteria in a dish or in a patient, and the effect that you see is there after 7 minutes, and the patient becomes healthy in a few days. So if the effect is very large, you don't need to do a large trial. And that may be very far away for ALS, but as I said, there's now one good result in a trial, a good first result in a trial in patients that have the SOD1 mutation. As you just heard from Amar, that's one mutation in patients with ALS. And this is, as you may remember this slide, part of it from high school, or I don't know, it's a DNA makes proteins, and DNA has genes, about 30,000, that's true, Amar, is it? Yeah, 30,000. And each gene makes a protein, and your whole body consists of proteins. So DNA makes RNA, makes proteins. And if there's a mutation in a gene, that leads to a mutated protein. And if that protein plays a large role in motor neurons, then motor neuron disease develops. So SOD1 is one of those genes, and there's now a therapy called antisense oligonucleotide therapy that actually is able to block, to treat the mutated gene. Actually, it's treating the mutated RNA, so that leads to a protein that doesn't have the mutation. Other kinds of gene therapies are being developed that are targeted directly to the DNA. This is one example of a relatively short trial, a phase 2 trial in SOD1 ALS. Phase 3 is ongoing, and as you can see, the dotted line. Look at the dotted line there. I don't know if there's a pointer. Yeah. Here, yeah, works. Is here. That's the normal disease progression. So it's quite a progressive disease. And in the treated group, the disease is almost stable. So that's a very exciting new result, and hopefully the result will also be found in the phase 3 study. So that's the first type of ALS that's going to be treated. And I think that's also what will happen in ALS. There's not going to be one treatment for all patients. It's most likely that ALS, motor neuron disease, will be treated in steps, in subgroups. Subgroups of patients are going to be treated. ALS may not be one disease, but maybe several phenotypes with different causes of the disease, and also those different subgroups may have to be treated with different kind of drugs or therapies. So that now I gave you an example for familial ALS, for genetic ALS, but 90% of the patients do not have familial ALS. So how do we measure the effect of therapy in sporadic MND, sporadic motor neuron disease? So in those for those therapies, for that kind of motor neuron disease, large effects are unlikely. There may be a slow effect, may take some time to see the effect of the drug, and to measure the effect of the drug, we need better, good outcome measures or biomarkers. So what is a biomarker? I think Amar or Matthew already talked about multiple sclerosis. That's an inflammatory disease of the brain. That's a good example of a completely untreatable disease in the 80s. Then the MRI scan was developed and became available. On the MRI scan, you can measure the number of inflammatory lesions in the brain. And so it's a very good biomarker to measure whether a drug has an effect. And 10 years later, the first drugs were developed, and now it's every year there's a new drug for multiple sclerosis. So biomarkers are very important. There's now one coming, neurofilament for ALS, but we need better biomarkers. Are small effects important? Is it important to discover drugs that have a relatively small effect? The answer is yes. So one example is HIV treatment. And well, this is the whole process of the HIV entering the cell, incorporating in the DNA, and then after a while it goes out and AIDS is started. It's a very short summary of the disease. And as you can see, there are many steps in the whole process where drugs are being developed, and they all have a small effect, but altogether they have a large effect. And now AIDS is a treatable disease. So if you go for the small effects and then add them all up, you have a large effect. The same is true for cancer. A cocktail of treatment has a much better effect, and it is important to discover the relatively small effects to finally reach the large effect. This is a multiple myeloma cell. These are all the arrows are processes in that cell where a drug is being developed. So how do we measure small effects in ALS? This is just an example of a functional rating, of the functional rating scale that we use in trials to measure the effect of drugs. And each line is one patient. So there's lots of variation between the progression of the disease between patients. So if you want to measure a small effect, it's quite difficult. You need a large number of patients. And it's also true that you don't want to have in your trials, it may not sound very nice for patients to hear, but you don't want to have patients in that do not progress, because you can't measure an effect in patients that do not have a progressive disease or do not progress rapidly enough. So you want to exclude those patients, and you also want to exclude those patients that have a disease that's too rapid to measure an effect. So what is the best trial? To actually have the best trial, it would be nice to have the machine they developed in the movie Back to the Future. Some people may be too young for that movie, if I look at the audience. Actually, they developed a machine that they could actually, Michael J. Fox, I don't know if people know, yeah, we still know him. He developed a machine where he could go to the future and see how it is in the future, and then come back to the present and change the future by changing something in the present. And so that would be the best trial design. If you would have, let's say 300 patients, you would go in the machine and look at them how they are after about a year. Then you go back to the present and give them a drug, and then see how they are after 1 year, and then compare whether the drug is effective. Yeah, that would be the ideal trial. Don't you agree, Matthew, Amar? Yeah. Okay, good. At least there's some agreement today. Yeah. It's good to agree before we take over Australia, right? I'll come back to that. Don't worry. So we need placebo-controlled trials, and that means that you have to have two groups. One a treatment group and a control group, and they have to be similar, otherwise you can't find an effect. So you have a treatment group, a control group, you do follow-up, and you compare them at the end. But at the beginning of the trial, you have to randomize, we call that randomize the patients to each group. that you end up with similar groups in the placebo group and the treatment group. Why is that important? I give you an example which is a little bit negative. It's not meant that way, but it just, I think, is important to show that it's important to do placebo-controlled trials. And one example is the diaphragmatic pacing. That was a device. It's a pacemaker for respiratory function. So it was put in the diaphragm, and it's actually you had to do surgery to implant it, and then it would stimulate the lung muscles. Is that explained? Yeah. And so it's very logical. It sounds very good thing to have. And the first study was an open study in 38 patients, and patients were measured, were followed up. That was the progression of the disease. And then at, sorry, this is Dutch. And then at 50% lung function, they would have the device. And as you can see in this open study, the disease course looked a lot better after the device. So that was very promising. And so the machine or the device was approved by the FDA because there was lots of pressure on the FDA to approve it because it looked very beneficial for patients. And the company that actually was selling it had a very aggressive sales style. I looked that up on Google Translate. That's when people that call at night and want to sell you something and they don't stop talking before you buy it. And actually that happened with that company. They would call again and again, and they wanted us to do surgery on our patients for this diaphragmatic pacing device. And so many patients were treated worldwide. But there were two placebo-controlled studies done, one in Paris and one in Sheffield. The first one in Paris, the red line is non-invasive ventilation and alone, and the blue line are the patients that have non-invasive ventilation as well as the diaphragmatic pacing device. As you can see here, the disease course is a lot worse in the patients that have the diaphragmatic pacing device. And the same results were found in the study in Paris. They did a slightly different design. They did a sham stimulation. So they implanted the device, but they didn't turn it on. So just to make the groups equally, both groups had the surgery, but one device was working, and in the other group it wasn't. And as you can see, the group where with the active stimulation, the disease course was a lot worse. So instead of 20 months better survival, it was actually a reduction of survival of 20 months. So it's very important to measure the effect of new drugs, new therapy in a placebo-controlled trial. So and that we have to improve, especially from the step from the phase 2 to the phase 3 studies, because the phase 3 studies are very, very expensive. And as you can see here, this is the percentage. It's not for ALS. It's actually not as good for ALS. The whole process here, from each process, only half goes to phase 2, only a quarter goes to phase 3, and half goes to the treatment phase. And if you add that all up, only 6% of the drugs make it to the market. For ALS, that's 1.5%. to 3%, depending if you count Edavrone. Is anyone from Mitsubishi here? No. So then it's 3, and otherwise it's 1.5. In Australia and in Europe, it's 1.5. So we have to do more, and we have to do better trials. 1 to 2 successes out of 65 studies is not enough. So we have to do better, we have to change. Only 3% of the patients are involved in trials. That should be much, much more. How many of you have been involved in trials? Yeah, that's not enough. So we have to increase that. And it remains a challenge how to measure the effect in a therapy. So we have to improve that too. We have to be careful for experimental therapy. I have to say this. Hope is not, a false hope is not hope. In my opinion, we have to inform patients better. What's on the internet is not always true. Some people think because it's coming out of a computer, It's better than what the doctor says, but I always say the better the website looks, the more careful you have to be. The other problem is that companies make unjustified claims on therapy, and we have to correct that as doctors. I think that is, well, I think it is really, a crime, I would say, how people take advantage of people with an untreatable disease. For lots of money, you can get all kinds of weird treatments that do not have an effect. But there is an effect claimed. So I think that's an issue that we as researchers, as clinicians, deal with every day, and we have to inform. We have to give our patients the correct information, although it may not be the information that patients want to hear. Very good site is ALS Untangled. I don't know if you heard about it. It's actually an organization, many of us are involved, that reviews alternative and off-label treatments in an objective way. There's a website, you should look at it. So what we want to achieve is more patients in clinical trials. We have to improve inclusion criteria. We have to use digital technology that's coming, innovative trial design, and less patients on placebo. So what we already achieved at most trials now have a 2-to-1 randomization. That means only 1/3 of the patients is on placebo and not half of the patients. We also have to accelerate the drug development process, and for that we need to develop a highway towards a cure. This is where we are now. We are every trial and any new drug. We are trying to invent the wheel again. We make the same mistakes again. That's also because the FDA and the EMA are quite conservative. I think better innovative design should be implemented sooner, and we want to be ready for all trials, and for that we need to have consortia. So we want to join Australia. We don't want to take over Australia. Amar, do you think so? We want to be part of PACTALS. Yeah. So Europe joins PACTALS, okay? That can be in the news. We do everything to find a cure for our patients. That's the message. In Europe, just to help the Australians, we try to develop a roadmap, which of course you already have, I'm sure. And that's, I think, the working groups that we want to establish. We want to see whether ALS is one disease. We have lots of data on environmental lifestyle risk factors. And on genetics, we have to develop better biomarkers, better trial design, platforms, trial consortia that should be very large. We can't develop a new treatment just in one country. You need many countries that join forces to have good trials, and we have to develop better outcome measures. I give just 3 examples on how we try to improve trial design and accelerate the process. One is that we developed a personalized prediction model of survival. So for each patient, you can enter 8 clinical variables that are all available at diagnosis and put them in an algorithm, and you can predict survival much better, the disease course much better than just the average. We developed that on databases from 14 sites in 9 countries in over 11,000 patients. So that's a very good tool to use in trials and to stratify better and randomize spaces better and to have better trial design. It's available online through your caregiver. So it actually helps to predict the patients that have a very slow disease course or have a very fast disease course, and to enter all the patients in trials that have the disease course that are best to find a new treatment. The other method we developed to accelerate the drug discovery process is to incorporate genetic data in trials. We re-evaluated all the lithium trials that were done years ago in the UK, Netherlands and Italy, and we found that one genetic variation, UNC13A, if you had that variation, those patients did respond to lithium in a post-hoc analysis, so that needs to be confirmed. And that's shown here. These are the patients that did not receive lithium. They had a faster disease course than the patients that were on lithium. And that we want to now confirm in a trial in Australia together with Europe. Is that good? Yeah, it's a good order, is it? Very impressive. I will stop doing it. Okay, yeah, we love you. We want to have lunch with pectals and ankles tomorrow in one big lunch together. Yeah, great, good. We pay for it. Okay. You can pay for it if you want. The other last slides are the platform design trials you have heard about. What it means is now, what we're doing now, we start a new trial. We needs to be a treatment group and a placebo group. We start 4 trials, as you can see here. It takes a lot of years to do all 4 trials, and if you have 4 trials, you have 4 placebo groups and 4 treatment groups. If you would now do them in parallel in a platform trial, you can take advantage of the placebo group. You only need 1 placebo group for the 4 different drugs. So that would be a lot faster, less patients on placebo. And what's the advantage? You can add on new drugs during the trial, and all those new trials on new drugs can also take advantage of the placebo group. Okay, last message is that we all should do this together. Patients, researchers, clinicians, also the ALS foundations and the pharma. We can't do it our own. We need each other. And well, all the work that we do is to find an effective treatment for all patients with ALS. We are going to reach that goal. We work very hard. We collaborate very well. We trust each other. And hopefully treatment will come very, very soon. Thank you.
Can you all... All right, now we come to the interactive part of the afternoon. So we've got just under an hour, so I'm going to call it quits at about 4:25 to give everyone a chance to have tea, coffee and network together a little bit. But we've got just under an hour to ask these world experts in motor neuron disease any question you want to ask. So we're going to run it. We've got a couple of roving mics. So if you could please in the room put your hands up. If you're streaming live on Facebook, we have 2 operators sitting with computers to take your questions and feed them to me. We've already got some questions from the floor, and I've got a couple here that have already been submitted online.
First of all, thank you very much for a very informative and also very inspiring discussion from all of you, so thank you. A question about possible causes. I'm wondering if diet is one area that you've looked at, and I'm just thinking about maybe areas such as gluten and other allergic type areas that have caused issues in many, and maybe those haven't done the action or changed the food, they've continued on. Maybe has that been an area that's been a possible cause?
So diet is an important and increasingly researched area, but it's an area that's only just started to be researched properly, so we don't have all the answers. Diet is probably important because it affects your gut bacteria. And your gut bacteria seem to be important because the gut in everybody, after they've eaten, for example, becomes leaky. And the bacteria in the gut come into your blood, and then your immune system is activated and wipes them out. And that immune activation seems to be something that might be important in triggering inflammation, and if it's not calmed down, then that might be a process that is involved in ALS. Another reason diet is probably important is once you've got the condition, it seems that a high-calorie diet probably is beneficial. We don't know that definitely because we need to do a trial to look at that, and that's in progress. But people who lose weight more rapidly seem to have a more rapid disease course, and that weight loss isn't just because of muscle loss; it's actually a fat loss. And that's something that actually the pioneering work for that was is done by Derek and Shu. Derek's in the audience there. So he actually can probably answer this question better than anybody else in the room. So I don't know if you want to take it over, Derek.
So thank you, Amar, for putting me on the spot. Our studies are very small, and we're conducting them mostly through looking at patients in Brisbane. So it's very, very early, those studies are. And so most of the work that I can report and talk about is really the literature searches. And so there are instances where the diet can be important. A great example is if you're deficient in certain components within your diet itself, and I think vitamin D has been shown to be quite a useful, vitamin B, sorry, is very, very useful in slowing it, but only if you have a deficiency. So I think what I really want to say is just caution to the wind. Yes, there's evidence to suggest that a high-calorie diet can be beneficial, but of course we need to know who's going to be at risk for losing weight and whether or not that diet is actually going to encourage weight gain, which could also have a big impact on them, especially if they have a disability, and the weight gain itself is going to add added complications to that disability. So as was suggested by Amar, it's something that we really have to research a lot more in order to provide people with the right advice on what to eat and how much to eat, and also who should be eating what.
Matthew, Leonard?
Well, I think it's a very hot area, and you mentioned celiac disease, so there's a significant increase in autoimmune conditions in motor neuron disease families. That's the first thing. The second thing is the whole sort of landscape of the gut-brain axis is changing. We're seeing that particularly in Parkinson's disease. Probably some ingested material is part of the process that's taken up through the stomach, through the vagus nerve into the brain. And then another sort of area, stream of research that's very topical and will be covered a lot in the next few days is prion disease, prion proteins and the spread of prions through the brain. It does look like there's a spreading mechanism. Obviously, this has never been proven for motor neuron disease, but I think Perth is a good territory because 20 years ago everyone thought peptic ulcer disease was due to acid production in the stomach. So when those conditions are discovered, it's a complete change. So I think you've got to have an open mind about the gut and its involvement in motor neuron disease.
Leonard?
Yeah, we asked patients to fill out questionnaires about their habit, diet, and diets before disease onset. And we found that patients have a higher calorie diet, but have a lower body weight. So that could be some evidence for hypermetabolism in patients. That could also explain the weight loss. And we also see that patients that have more Mediterranean diet, so more vegetables, more fruit, that seems to be a little bit protective for ALS.
So we should all go and live in Italy.
Yeah,
Yep.
that's a good option, yeah. I like Perth too. But
We've got a question up in the back corner.
Yeah, I had a question around Leonard's presentation on clinical trials. And I think you proposed kind of the ideal moving forward clinical trial design or what's needed. But what I didn't see up there was really the creation and the use of biomarkers and how important that would be so that we can move away from ALS-FRSR as a way to predict or gauge success of a trial. And there was no mention of even within a single patient, the rate of progression can change over the course of the disease. And I don't know that that's addressed at all in the current clinical trial design as you presented it.
yeah, the ALS-FRS score that we use now needs to be improved. I think we all agree. So we're working on that. We don't have results yet. We're all working on biomarkers, better biomarkers. They are going to be implemented in the future. The neurofilament, I think, is a new discovery that's now also, but it's a biomarker for neuronal loss, not for a specific pathogenic ongoing mechanism. That's so it is a good biomarker for neuronal loss that is going to be used, and it's also actually was also shown in the SOD1 trial that the neurofilament also would go down if the patient improved. So I think that's a very important new new biomarker that's probably going to be implemented in all new trials from now on. But I agree, we need better biomarkers, and we're all working on that. That will really shorten the trials, I agree.
I also think that there are now incorporations of rates of progression in clinical trials, although a lot of these have been done post hoc analysis. They're looking at the delta, the change of progression in patients. And some of the more recent medications that would include both masitinib and edaravone have moved forward by saying that they have a greater effect on the faster progressors. I mean, but this has been done as a post hoc analysis, so I think this has to be incorporated more at the beginning, the planning stages, as much as what you're saying.
The other issue, of course, is that motor neurone disease isn't a static thing, as you pointed out. It's something that changes. And having a biomarker that monitors that change actually may be not what you need. You might need a marker that just tells you, is the disease process active, and then how much can you switch it off? And that's not necessarily the same as a disease progression biomarker. So actually the biomarkers that we've got and that we're developing is a really challenging big area. And I think Martin Turner will be talking about that actually in the conference.
Got a question here in the middle of the room. Yeah.
Yes, I had a question for Dr. Al Chalabi regarding the lifetime risk, the 1 in 300 by age 85. I presume those numbers were done with the European population. Had they been replicated in a non-European population? And if so, what would those numbers be?
So it depends what you mean by non-European population. They've been replicated in the USA, but of course that's a European-derived population, largely. I don't think they've been done in a non-European derived population, but they have been replicated in other places. A good way of checking the kind of, without doing the formal study, of just checking the rough level, which you could do in any country actually, is by looking at death certificates. If you assume that death certificates are relatively accurate, which they're not, but that gives you a rough rule of thumb of what it would be. So if you look in the UK, for example, about 1 in 300 death certificates has motor neurone disease on it. So it does match.
But it's a little bit lower in Latin American population.
So obviously the average age of the population is going to affect it. If you've got a younger population, then it'll be a lower risk as a lifetime, because the life expectancy will be lower.
Any other comments on that one? We've got we'll take a question from Facebook live streaming. Thanks, Morag.
Right, this is a question from Dr. Sain from India, who unfortunately couldn't be with us as she was unable to get a visa for Australia. So her question is, what are your thoughts on copper?
Matthew, I might pass that. No? Yeah.
Yeah.
Well, look, I think for a start, it's good to see that there's interest in clinical trials coming from pharma companies. And CUATSM is an agent that has been supported through philanthropy from Australia. It actually started off as a ligand that's used for PET imaging of the brain, and so it was then patented through the Florey, and the first phase 1-2 studies have been undertaken in Australia. But I suppose the jury's out, and what people need to do now is do a proper phase 2 or phase 2-3 study, much as Leonard has suggested, placebo-controlled, because there can be a degree of sort of, everyone is positive, so patients are positive and clinicians are positive, and then when you get to the end of a trial and you see there's no effect, you wonder what that positivity was about. So I think you have to just do it properly through scientific channels.
We've got another question in the middle of the room over there.
So this question is more about clinical trial design in general. So the edaravone trials have shown the benefit of enriching for a specific population within the trial design, in that they go from showing no significant effect to a significant effect when they select for that trial, which I think highlights the importance of heterogeneity within a population. How feasible do you think this is going forward in clinical trial design, where you're trying to specify or enrich for a certain population where it might not be highly prevalent, and that you'd have to have a longer period of enrolling patients, which may make the trial design more difficult overall?
Yeah, I think I think the design is a very nice design in the sense that it was designed based on a post-hoc analysis. So they first did a large study in 300 patients, no results, and then they did a post-hoc analysis and found that the subgroup may have an effect, and did a second study to confirm that. So that whole concept, I think, is very important. I think that's one of the lessons that we should learn, that once we do a trial, we should keep the data close to us to actually to use the data for future developments of new treatment. In my personal view, and also, no, I shouldn't actually mention my personal view. I should mention the view of the EMA, so that's the European FDA. They think, or their opinion is that the duration of the trial, so 6 months, is too short to approve the drug in Europe. And we, as European centers, sent out a statement that we think the company should have done or should do a trial with a longer duration of at least 12 months to have some indication what it does on the long term, but also what it does on survival. So I think many companies are copying the design of the latest Adavron trial, and I think they forget that it is a post-hoc analysis, that it was a post-hoc analysis.
The other thing to know is, unless you know the mechanism of your drug of action and can pull out the subgroup of people who that might be important for, then doing a subgroup type of clinical trial isn't going to be a very easy thing to do. So we can see that with the SOD1 antisense trial that Leonard mentioned, where you can easily pull out from the mechanism of action that you know, which is to do with SOD1, and you know the people with SOD1 mutation are going to be your target. So you can do that, and you can look for a large effect even though you've got a smaller population, because your disease mechanism and the subgroup you pulled out match. The difficulty is if you don't know that information, which for most cases we won't, in which case the only way forward is the way Leonard has just said, which is you do a post-hoc analysis of a trial in everybody, pull out the subgroup that seems to have an effect, and then repeat the study in that subgroup of people. In fact, that's what we did do with the lithium study. So the lithium study that was shown, again by Leonard, where people with UNC13A genotype that normally gives you a bad progression, if you give those people lithium, it seems to improve progression. That's a post-hoc analysis, and so now it's being funded for a phase 3 study to confirm that effect.
So I'm just going to post a question that I'm always asked in my clinic, and I'm very interested in all of your replies on this. Why can't or doesn't stem cell therapy work? I want all of you to please comment.
Well, I think that is an important question. So that's one of the first questions that is asked. And I think that it's an emerging area of science, and it brings a lot to the table. The reality is that there aren't too many trials that have been taken through in appropriate centres under scientific guidelines that have shown a benefit. That's not to say that won't happen. And so I said there was already one released today, and then will be others that are coming. And I think that this is an emerging area. I also think, though, that it's unlikely that you can rebuild an adult sort of neurology system, so the brain and the spinal cord. That doesn't mean, though, that you can't put cells in the environment that preserve the function of damaged or deteriorating motor neurons and other brain cells. So I think there's a number of different areas that people are looking at, and it'll be a massive focus of the meeting. So we'll probably learn more over the next few days.
So my view on that is that stem cells, the way most people want stem cells to work is that you'd put them into the nervous system and then they'll replace the lost motor neurons, that they'd grow and reconnect and rewire the nervous system. But that's almost impossible to achieve in practice because the nervous system is so highly connected and the nerve cells are so long with such a path. I'm going to try and do this Australian scale. If you had a nerve cell that was about 30 yards across, then the motor neuron axon, the actual wire bit of the nerve that connects to the muscle, would be like a tunnel going from Perth to Auckland. So it'd actually be a little bit further than that, about an hour's flight more than that. So maybe down to the South Island. So they're very, very long, and so it's a meter long typically, a motor neuron. You're asking the new stem cell to grow a wire into an existing nervous system 1 meter and find the target, which is very difficult to do, and at the same time be connected in all the other places. And it's a cell that doesn't know that body at all. Whereas when you're growing, you start off as a very small object and all your nerve cells are connected and then they grow with you. So the scaffolding is there and they know how to connect. So trying to regrow an adult nervous system is very difficult. So the only feasible way at the moment to use stem cells is to assume that they're going to secrete some kind of cocktail of substances that will keep the other motor neurons healthy around them. In which case they might work, but actually I would argue why not find out what that cocktail is and just give it directly.
I agree with Amar and Matthew. I just want to add that I think stem cell therapy doesn't have the antibiotics effect, so it doesn't stop the disease. Some clips on the internet suggest that people start running around. I know some of their physicians, and they say that the disease progresses afterwards, and people don't put that on the internet when it progresses. So I don't think it stops the disease. So placebo-controlled trials need to be done also for stem cell therapy. Some people say that it's unethical to use placebo in that kind of treatment because it's an invasive treatment. You need surgery, spinal cord surgery, so it's not ethical to use placebo. I completely do not agree with that because I think the more invasive the treatment is, the more important it is to do a good trial to show whether there's an effect, because otherwise people are going to be on surgery forever. So I think it's very important to very carefully measure the effect and also look at the burden of the treatment and the effect.
And I'll just add one more comment. The analogy along Amar's line in terms of regrowing motor neurons that I give my patients, it's like taking a computer and unscrewing it. You pull out half the wires, go to the local shop, buy another whole lot of wires, just throw it at the computer and screw it closed and hope that every wire connects to the exact right thing so your computer works again. We're just not at that level of technology to be able to connect this bit of the brain to this part of the body yet. We've got another question up the back. No? Okay, another Facebook question then. Please think of some questions. You've got a real opportunity here with world-class experts.
This is a question from Callie. Could antiretrovirals be useful for all ALS patients or only a subset that are HERV-K positive?
Should we answer that? Yeah. So in the initial study that I showed you, where 60% of people have a retrovirus signal in their blood, that doesn't mean that only 60% of people will ever have a retrovirus signal in their blood. We've done some other studies more recently that suggest that actually the retrovirus signal comes and goes in everybody. And so just if you do a cross-section of people, then about 60% at any one time will have it. So it's likely that if antiretrovirals are effective, and that's a big if, we've still got to do the trial, but if they are, then they're likely to be of benefit in most people. There's no reason to think they'd only work in people who have that retrovirus signal. It may be that eventually we can pull out a subgroup that does, but at the moment there's no reason to think that.
Any other comments? I think I saw the... Is there a question over there? Right over to the side.
Hi there. Has there been any proof that medicinal cannabis helps the neurological system with this disease?
Yeah, well, I mean, a lot of patients are on medicinal and non-medicinal cannabinoid therapies, and it is very effective in managing a lot of the symptoms of motor neuron disease. No one has done a trial to look at effects on disease progression, but I suppose it's a really emerging territory, and the typical sort of cannabis approaches that we may think about, that's only one part. There's about 9 variants, and I think this is, again, a science that's developing and evolving. But at the moment, I think all you could say is very useful for certain symptoms, no clear science suggesting effects on disease progression.
Anything to add? We've got a question here again in the middle of the room.
Okay, so this is for any of the 3 in the front. So normally we have very animated discussions during research visits with patients, but the other day the conversation died very quickly. It was a patient with a family history, and they asked us whether or not they should select against MND when they go for IVF. Do you have any opinions on that?
That's an option. It's a choice. I mean, I think, yeah, it is possible. So that's an advantage of genetic testing, that actually you can select an embryo that doesn't have the mutation. So that could be a reason to do genetic testing.
So I would ask which variant they carry.
Yeah,
if it's a variant that doesn't have a high penetrance, in other words, it doesn't necessarily mean you're going to get the disease, that might be a more difficult question. But if it's, for example, the A5V variant of SOD1, we know if you carry that variant, you're very likely to develop a very aggressive form of ALS. Then that probably would be a reason to consider an IVF approach where you're selecting not to have that variant.
It needs genetic counseling, of course.
Yeah.
Carefully done.
Yep. I mean, so we actively do that pre-implantation genetic diagnoses, and I would say, you know, the families and the individuals are very happy. It's not really up for us to make the decision, but if they want to do it, of course we're going to support it. And taking a step back, if you could have that power that you wouldn't have to go down the pathway and have that hanging over you, I would say it gives people a lot of peace of mind.
And it's important to say you can actually do it without the person, so anybody can have this done without even knowing their own genetic status. So if you have a family history, but you know the gene in your family, but you don't yet have symptoms, and you don't know if you're carrying a disease gene, you can still have the fetus tested, the embryo tested and selected without knowing your own genetic status.
Once gene therapy is coming, that whole kind of debate will change substantially, because then it becomes a treatable disease. It's most likely that there is an option also maybe for prevention of carriers. So it may become more important to know how your genetic profile is in the future. These kind of questions will come.
I'd be interested, Merrily, what do you do?
I also give my patients the option, and I explain to them what the options are these days with pre-implantation diagnosis, and whether they themselves want to even find out with predictive testing, and then I send them to genetic services for the genetic counselling for them to make an informed choice. Yep. I have a sort of related question that came online, so it's again related to gene therapies. This says, of all the potential therapeutic approaches and technologies right now in 2019, antisense gene therapy appears to be emerging as a front runner and seems to have legs to complete the journey. Should we seek to accelerate research and investment into this area as a matter of priority globally? And if so, what are the urgent prerequisites for doing this as expediently as possible? Thoughts?
Do you want to start?
Yeah, I think for the SOD1 now, it's a reality now that patients, there's a shortage of patients. So we need better databases and more patients. So we need to involve Asia as well, and other continents that are maybe less involved now, to actually have sufficient number of symptomatic gene carriers to do these kind of studies. I think that now, whether the same effect will be found in the C9orf needs to be, of course, investigated. But the hope is there.
Well, I suppose it has been prioritised in the fact that, like, this week, a number of companies are coming here to try and organise their various approaches. So I suppose that just shows from a business model approach from industry, pharma, they think it's worthwhile, and that they're the ones who can drive this process. So I think we'll see a lot of developments.
And it's important even for people who don't have a family history, because up to about 14% of people in the UK, for example, and probably in other parts of the world, have a gene variation that has a big gene variation that would otherwise cause familial motor neuron disease, even though they have no family history. So actually gene therapy might be important for a very large number of people. Maybe 1 in 6 people could be treated.
I think it's such an exciting field, seeing human trials now being done in the SOD1 patients and C9orf just on the horizon. And if you look at other neuromuscular disorders, a related one, spinal muscular atrophy, you know, there's a gene therapy there that's a potential cure. So I think there's genuine hope. I agree.
We've actually just started. We've just given our first C9orf72 injection.
Oh, fantastic. Yeah. Oh, that's exciting.
So what's the policy in Australia on genetic testing? Also, do you give the option to all patients, also sporadics?
So it varies from centre to centre, so we'll hear what Merrily does. Because I think, well, C9 is, in our Australian population, present in about 6.5% of sporadic ALS, we test, or we offer the test to all patients.
Yeah, so do we.
I too discuss it with all patients because of the sporadic numbers that are positive actually for genes even without a family history, and then I let the families make decisions because of the implications on insurance and on family members. Some patients decide not to test and others are very keen to test, so I leave it up to them.
Just it reminds me though of your 4 footballers. Were they related?
Not as far as we know, but they were never tested like that.
Right.
We had a question I think down the front here. Sorry, who's got the microphone? I can't see. Morag, okay. More Facebook questions.
Yep. Another Facebook question. So this is from Anne. Is it still useful for someone who has been living with MND for 12 and a half years to continue to take riluzole?
That's for you, Amar.
So I don't think anyone will really know the answer to that. The evidence, if you look at all of the databases and do the analyses, which Leonard and I have done, and I think Matthew has done as well, shows that riluzole seems to have its biggest effect at the start of the disease and at the end of the disease, but probably does have an effect actually in the middle part as well, but to a smaller extent. So in those studies, we didn't make any assumptions about the duration of the disease. It was done by clinical staging, which we haven't discussed today, but it's something that happens in cancer where you stage the disease and you say stage 1 is early disease all the way up to stage 4 or 5 is late stage disease. You can stage motor neurone disease the same way, and you can show that riluzole has an effect in stage 1 and stage 4 that's biggest, but does also have an effect in stage 2 and 3 that's smaller. So it doesn't matter necessarily that they've had the disease for 12 years based on that kind of evidence, because we didn't take into account disease duration. So nobody really knows, but my guess would be probably it is still worth it. The other thing to say is that the original clinical trial of riluzole showed a 38% improvement in survival at 1 year. That's why people say it gives you 3 or 4 months improvement. It's actually not 3 or 4 months improvement really, it's 38% at 1 year, because that's all you can say. But the method used, the statistical method used, is what's called a proportional hazards method. And that assumes that the effect continues regardless of how long you take it. So that would suggest if you've had the disease for 10 years, then you've taken riluzole all that time, you get a 38% improvement in survival on average over that 10-year period, which is another 3 and a bit years. So you could argue that if you've got slower progressive disease, it's better to take riluzole.
In my experience, it's safe. The long-term safety is not an issue. I mean, I've had many patients that have used riluzole for years without any problems. So personally, I would say if you don't experience any side effects, I would just continue using it.
Yeah. If it ain't broke, don't fix it. Yep.
Yeah.
Have you got any other? Oh, we've got one down the back, sorry. There's a hand up. Yep.
Thank you. Kathy from the US. Can you speak to delayed diagnosis? When we looked at the slide on the poor uptake on clinical trials, I know in the US, by the time many people are even diagnosed, they're out of the window for eligibility to a trial. So,
yeah, the prediction model that we developed could solve that partially, so that is not a fixed disease duration, but it depends on those 8 variables in your prediction model whether you will be included in a trial. So that may help to enlarge the number of patients that are eligible for trials that can be included.
The other problem for clinical trial eligibility is many trials require you to be able to swallow for the duration of the study. And that's because if you change the formulation so that it's a form that can go from a tablet into something that's then crushed in the trial, technically that's a different formulation and therefore wouldn't be part of the same trial. So the only way around that would be if companies made things that don't depend on you being able to swallow easily, such as a liquid. Even then, administering it through a PEG tube, for example, you'd have to know that it doesn't stick to the plastic of the tube. So, but it's not an insurmountable problem, and I think that's something they should also concentrate on, because otherwise that excludes automatically about 25% of our patients.
And I suppose with the advent of more clinical trials, it's inevitable that the diagnostic delay is going to go down. There's going to be far more, I mean, I wouldn't call it a positive diagnosis, but clinicians will be more likely to make the diagnosis. I mean, in the environment, say 40, 50 years ago, where there's nothing that people could offer, there was perhaps a nihilism and a suggestion of why come up with a diagnosis. And I think that whole environment is changing very actively, driven by patients, families, and also by having dedicated clinicians working in the field. But the diagnostic delays are going down.
It is a very difficult journey, I think, for patients. I think Drew alluded to it earlier. They were the most difficult months. And if you want to tick all the boxes for some of the diagnostic criteria, you can wait many months or even sometimes years to tick all those boxes before perhaps some clinicians are perhaps sure enough to say that it's definitely motor neuron. But I think, as Matthew said, there's a reason to diagnose people earlier, even if it's in one limb these days. I think people are more often receiving that positive diagnosis or getting referred to the right people who will make that diagnosis more confidently.
Also, I think there are criteria for enrolment in clinical trials, and that's different from diagnosis. So you might be forced to go down to certain criteria in a clinical trial environment by the sponsor. And I suppose taking that also further ahead, there's a meeting tomorrow of the World Federation of Neurology, and there was a meeting earlier on in the year about coming up with diagnostic consensus guidelines that would actually try and streamline the engagement of patients in trials earlier. So that's something that I think everyone realises that needs to be improved.
All right. Feel free to keep putting your hands up so the microphone people know where to go for questions. Oh, great. Yep, up the back here.
Hi, can you comment on the reliability of the NCALS predictive tool and what, if any, are the barriers for it being used more widely in a clinical setting?
So it's quite reliable for patients that have a average disease duration or a short disease duration. If the prognosis is better, the interval, the confidence interval becomes wider. So then the prognosis, it's more difficult to predict an 8 or a 15-year-old survival than a 3 to a 5-year-old survival. So that's part of your answer. It is available through the internet to caregivers and not to patients yet. And because we talk to ethicists whether it's actually ethical to, because it could be a disappointment if it's shorter than expected. So we did not make it available for patients yet. We are investigating that now in a proper way. We actually doing research on that, how people react and whether they regret that they have done the test. So we're a little bit careful to make it available to the public yet.
I would also say it's not giving you a single number. It doesn't say you will survive this long. It gives a probability curve for the likelihood of survival to a certain length of time. Yeah. And so there's always a probability that you'll live a long time or a probability you'll live a short time, regardless of what the actual middle part of the prediction is. And I think it's important to bear that in mind, and most people can't really understand probabilities very well. A way of thinking about it, which I always tell my patients, is that I can make a very accurate model that will predict the survival of the light bulbs in this room. And I could come back in 5 years' time and count how many light bulbs are still there, and I'd be correct because my model's nice and accurate. But I wouldn't be able to tell you if that particular light bulb will be there in 5 years. And it's the same kind of problem that a survival model does. It's useful for selecting people for trials, because on average it'll work. But for an individual, it's not necessarily so useful.
It's a really important point. Are there any other Facebook questions? Yep, let's go to Facebook.
All right, so it's a question from Carly. What clinical trials are currently underway in Australia?
Well, the genetic therapies are underway, so the SOD1 study nationally, and then C9 will kick off in the first quarter of next year. I'm pretty sure the CUATSM is recruiting at present, and there's the refals and cytokinetics recently finished. And there's also going to be a nano gold study that will kick off perhaps before the end of this year. They're the ones I know of. Do you know?
Yep.
So the Julian Gold study should be kicking off through the European and Australian partnership, depending on what Leonard thinks these days.
Partnership.
But that should, Leonard's convening a meeting this week, and we'll hear more about that. Just back to his other stuff, I mean, the NCALS model, I think, is a very important piece of work, and I think something that Leonard has driven himself with his group and now through NCALS, and I think it'll be a very powerful tool and will help streamline the clinical trial approaches, and I think that's an example of the innovative approaches that people can come up with using the technology of the day. The other thing with biomarkers that we're struggling with is we don't have too many positive clinical trial agents, but we'll have like the first session on Wednesday is Martin Turner giving an overview of biomarkers, and there'll be whole sessions on biomarkers. What we really need is 1 or 2 therapies to come through to refine the biomarkers to a greater extent.
There's a question down the front here.
In that sense, it's an exciting time that many companies are interested in ALS. So it is, I think it's somewhere on the list that it could be a treatable disease in the future. So I think that in that sense, yeah, we're all very excited.
I was really interested in the discussion about personality as being a possible cause, and when you think about people like Drew and my father-in-law and many others that all of us have come into connection with, it tends to get the best of the individuals. And I'm just wondering whether it should be the role of parents to bring up our kids differently. We should be treating them as, like, they should be nasty, they should be physically inactive and medically unfit and so on. I mean, it'd be interesting to explore that a little bit more, wouldn't it, the personality side?
We should do a randomised trial.
Observational might be more ethical.
In our study, we found that patients with ALS are actually otherwise healthier than the control group and fitter. So in that sense, it's difficult. You become in the area whether you want to have cardiovascular disease or ALS. I mean, that's a stupid discussion. Yeah. Yeah.
And I suppose everyone in this room is a sort of product of tens of thousands of years of evolution. So everyone here is a success story. And then you say, well, why would motor neurone disease exist? And I presume there were certain evolutionary benefits that people saw from this. And people talk about, running till you drop. So having that sort of athletic approach, that was something that probably, when people were dying at the age of 35 or 40 standard, that you'd want these people, these key sort of individuals as part of your group. And I suppose the C9orf gene has come from in the majority from a single sort of Finnish person of mutation about 1,000 years ago. And then we can see that sort of spread through Northern Europe, through Viking spread into North America, down to Australia. It didn't really go into Asia very much because they had the Taipan dynasty and they had their own great warriors and they got rid of the Europeans. But you can see that that this is what society wanted. And now we're seeing the flip side of that as people are aging, getting into 50, 60, 70, 80 more often, that we're now seeing neurodegeneration playing out more commonly.
There's just a question in the middle of the room. Yeah.
Yes. Thank you. Look, my question, I'm Ruth from Mnd Association of Victoria. My question to the panel is, with footballers being at seemingly increased risk of MND, what's the panel's thinking on concussion and brain injury and the link with MND?
So people have looked at whether traumatic brain injury increases your risk of MND. In fact, a study has just recently come out suggesting that it might, and actually also might increase your risk of other neurodegenerative disorders. But if you look pathologically under the microscope, people with traumatic brain injury that's chronic can develop the same types of pathological change. They can develop TDP43 accumulations in the same way as people with motor neuron disease can. So that might be a mechanism, but it's not necessarily people who head the ball who get it. And people who box, for example, who are getting repeated head injuries are actually at more risk of Parkinson's disease rather than motor neuron disease. So it may not be that head injury is relevant. It might simply be that being athletic is relevant. And as I said before, just because it's associated doesn't mean it's causal. It might just be that if you've got an athletically tuned body through evolution, that's what's predisposing you to get motor neuron disease.
Yeah, well, I think the study that Amar was talking about was in the New England Journal of Medicine about a month ago, showing a significant increase in motor neuron disease linked to CTE. I think this is it's a controversial area that does need a lot more research. So I think the jury is still out. The other thing is we're talking about chronic trauma causing TDP43, so the pathological signature, we still don't fully know that TDP43 is causative. So is that an effect or is it causation? So a bit like, you know, a fire going through a forest, the fire sort of passed and then you're left with the charred remains, and that's the sort of TDP43. Was it the TDP43 spreading that caused the process? And I think more and more I would say it's probably an effect, but it's a marker.
Any other comments on that?
No.
Jeff?
Yeah, just wondering, we spoke of it before, but is there any link with the way our food is being produced and chemically, we're not picking it from the trees when it's ready, we're you know, it's growing in a fridge and it's, you know, all those genetic changes to our foods compared to the caveman that used to go out and hunt and get it real.
So I don't think there's been a study of processed food versus unprocessed. Leonard might have done it in Neurodegeneration. No. So I don't think you can fully answer that with the available evidence. Processed food versus unprocessed, we don't know.
Yeah. Facebook question?
It's a big question. It's from Ross. How many years do you think it will be before we have a cure?
It could be next week, or it could be another 10 years. We don't know. Sorry.
I think it depends on what you mean by a cure for everybody, or a cure just for a proportion of people. Because I think the 2% of people who have SOD1 mutation, antisense therapy is around the corner, and we may have a cure in a year or less. If you're talking about a cure for everybody, that's a very different thing.
Yeah, well, I think the genetic conditions are going to be cured a lot quicker, and I think that it wouldn't be unreasonable to think that within the next 5 to 7 years there'd be more effective therapies generally available.
Yep. Any questions in the room before I read out another one from online? So this is a wordy question about collaboration, really. So we've sort of discussed it a little bit through your talks. It says, this event shows the need for and the power of international collaboration amongst academics. Big Pharma is, of course, global also. Given the commercial needs of the major pharmaceutical conglomerates, question 1, would it be possible to put them together into a global academic and research strategy that might persuade and enable them to accelerate development once prime targets are identified? And 2, what might that strategy look like? So I think some of you addressed that in your talks, but...
These kind of meetings are very important. So we talk to each other all the time. It's the busiest week of the year. It is extremely important to collaborate. We do collaborate a lot, and so I think if anyone worries that we don't collaborate, that's actually not true. But sometimes there's also some competition, which is also good, because that makes us work a little bit harder either, and even our PhD students work harder. So some competition is not wrong, and sometimes you need replication. You need to have the same study, two centers in the world, confirming each other. That's actually very important in research. So you don't need to do everything together, but for some areas like genetics, also biomarker studies, trials, you can't do it on your own. You can't do it just in one country. You probably can't even do it in one continent. You need to collaborate to get good results, and we do, and we need to encourage that, and we also need to encourage that the foundations collaborate with each other, because then that helps us to collaborate as well. If the funding is, but on the other hand, we don't need one funding thing for the whole world. I think, I don't know if you want to add to that.
Yeah. So I would completely agree with that. We already collaborate very strongly together, and that is essential, because without collaboration you don't get the numbers. I think there are 2 types of researcher. There are the types of researcher who like to keep everything themselves, and the type of researcher who doesn't mind sharing the information. And if you're part of the general public, you might say, well, why doesn't everybody just share their information? The reason is we're measured by our universities by the number of publications we make and where we are in terms of the publication position. And we're measured by the grant-giving bodies that give us the funding to do our research by our track record, which is the same thing. And so if you share your data, you're effectively diluting your prominence in the field potentially. And I think what we have learned, definitely the three of us on this panel and others, is that by collaboration you actually boost your output, because you get a more powerful result because you've shared your data with other people. And not everybody has fully understood that, but I think that's a general trend across the world in research in motor neuron disease particularly. We're actually a very collaborative community of researchers, and we do work very strongly also, of course, with patients and patient foundations, and actually also with, we're beginning to bring pharma on board. There's always been a reluctance because pharma is commercial and we're the opposite of commercial. And there's always been a kind of conflict ethically and culturally and all of those kind of things. But we are beginning to bring pharma on board much more and work globally with them.
And I think one of the key drivers has been this international symposia. So meeting up over the many years, coming to meetings and hearing each other present results and how to go about it, that's been a learning experience, but it also then drives friendships. Friendships to the extent that Leonard sends his daughter to come and stay with me for about 3 to 4 months on a one-weekend visit to Sydney. But
turned out that Matthew is a better father than me, so I hear that every week.
But
he
I
does
think
barbecue on Friday night, my daughter said. I never do. I hate barbecue.
Yeah, I think the consortia and the meetings are fantastic. I think we've always wanted international pharma involved too, but it's been an area that they didn't want to invest too much in. But and that's why I think the community awareness, the Ice Bucket Challenge, and the philanthropy that is there to bring trials to Australia, to get Australians to collaborate with Europeans and North Americans. And then we can also see the power of improving the standard of care for patients across clinics to say, well, this is the model of care that ideally we'd like. But you have to have some sort of vision of where that is. And that only comes through the interactions at national or local, national, international meetings. So I think this symposium is a critical part. And the things that are going to happen both are sort of covertly and clearly, and in breakfast meetings and dinner meetings, and all of these events are critically important to basically accelerating the path to treatment.
Yeah. Can I just add one more thing, which is that the other sea change that has occurred in our collaborations is the involvement of patients in actually the design of the research and in which questions need to be answered and how we answer them, and how should we do a trial, how should we do the research, is it acceptable the way we've designed it? That's a new thing that's happened in the last few years and is increasing.
Yep. It's important that we measure what you guys think is important. There's no point us measuring a change in strength if that's not what's important to all of you. I think we'll just take one final question from up the back, if that's all right, because we're just getting close to closing.
So I just locked in. Blue-green algae's been in the media in recent years as a potential cause for MND. Do you know if there's any research going on regarding that topic, and what's your personal views towards it?
So blue-green algae are supposed to produce a toxin, a cyanide-related toxin. So there's a kind of theoretical reason why they might be important. There is, if you look in the literature, there are a few examples of outbreaks of motor neuron disease-like conditions that have occurred when people have consumed mussels that have been where there's been an algal bloom. So it's not impossible that they might be relevant, but those kinds of examples are motor neuron diseases that are static, a bit like polio. So once you've got it, it doesn't then progress in the same way as normal motor neuron disease. I'm not aware, if you look at those location map type studies like the one I showed you, there's none that necessarily accumulate around the coast or in places where people eat more blue-green algae, things that have eaten blue-green algae.
Terrific. Well, I'm just going to make a few closing remarks and thanks. I hope that you all really appreciate what a fantastic session this has been for all of us. I first of all want to thank the International Alliance of the MND and ALS Associations for hosting and sponsoring this session. To the volunteers and the organisers from Motor Neuron Disease Association of Australia, and in particular I want to recognise Carol Burks and all the amazing work that she's done to pull this together, as well as Morag and Ben here who have been fielding the Facebook questions. Thank you very much. And from the volunteers from MNDWA who have been running around all afternoon in all the sessions, but here with the microphones, that was fantastic. I want to acknowledge the courage shown by Mr. Drew Bathgate to come up here on stage and open with his story. I think it really set the tone for a fantastic session this afternoon. Then we opened with Professor Matthew Kiernan, who emphasised again the importance of collaboration. And if you haven't picked that up this afternoon, then you need your hearing checked. Matthew emphasised the importance of collaboration not only for research but to improve our understanding of the disease, but also to improve the treatment of the disease and really to standardise it across not just Australia but the world. And he talked about the new trials that are around, and I think provided really some really serious hope for the future for all of us here. Professor Al Chalabi discussed the mysteries of motor neuron disease, and in particular talked about the 6 steps to developing motor neuron disease, and the genetic factors really account for 3 of those steps, but then there is another 3 steps or another 3 hits that we don't fully yet understand and probably going to be different for all sorts of people. And then we finished with Professor Vanderburgh from the Netherlands, who helped us understand the phases and the processes of clinical trials and some of the difficulties we face when designing clinical trials. He talked about the back to the future model, which would be the best model for clinical trials if we could really do that without having to put people through the placebo arm. But until then, the importance of having that placebo-controlled arm. That's often a question I'm asked when I'm randomising patients for clinical trials. But I think it's really important to understand that if you're in the placebo arm, it's just standard care. It's not that you're getting nothing, you're still getting standard care. But you may or may not get the active drug. But hopefully you will get it at the end of the trial if it's positive. And really we need to aim to involve as many patients as possible in clinical trials, and we would ultimately aim to offer clinical trials to all patients as they come into the MND clinics. So I think that's been a fantastic session. You've asked some wonderful questions. I think the panellists are probably all in need of a drink, I think, to recover. And I would invite you all to stay and mingle and network over afternoon tea. So thank you once again for your attention and your participation in this session.